Hallo.
Zwei Vorträge vor dem Ende des heutigen ersten Tages. Wir bisher von Trennung gesprochen haben, dann sogar eher von Quervernetzen, das heißt also Schaffung größerer Einheiten, geht es jetzt in den letzten beiden Beiträgen um das Schaffen von kleineren Einheiten aus Proteinen, das heißt mit Zerlegen, also ähnliche Richtungen ermöglichen Verarbeitbarkeit oder Veränderbarkeit von Proteinen. Und die nächsten beiden Vorträge stammen aus einer Nachwuchsforschergruppe, geleitet von Dr. Cheison am Ziel am Zentralinstitut für Ernährung und Lebensmittelforschung. Und da ging es eben in den letzten Jahren um Entwicklung einer Prozessplattform zur Steuerung der Hydrolyse von Proteinen. Herr Cheison wird einleiten und dann der zweite Vortrag von Frau Nehme, die sich mit einer Teilfragestellung zu diesem Projekt befasst. Bitte schön.
Nee. Thank you. Excuse me if you will, we switched the language slightly for obvious reasons. As Professor Kulotzik has said, my names are Cheison, Seronei, there are very many. And I come from Kenya originally. I went to school in China. That's where I did my master's and PhD. And then Professor Kulotzik was kind enough to offer me an opportunity at his institute. And that's where I've been for the last 6 years. What we do, basically we work with proteins and enzymes, so digestion of proteins using enzymes. And my colleague Elena Leib will tell you about peptides or directed hydrolysis of proteins to get specific peptides. But I'd like to mention to you what we do to use enzymes in purifying proteins as a prerequisite to use, for example, of membrane technology. Let's go to a brief background, which is what you know, that in milk you can separate milk using centrifugation to get skim milk on the one hand and cream on the other. And from skim milk, you can further fractionate the proteins in skim milk using several methods that are available. You can use chemosine, for example, which attacks kappa casein to coagulate caseins, so you can separate the casein micelles, which are insoluble on the one hand, and whey proteins, which are then soluble on that by using chemosine. You can use isoelectric coagulation, where you can get casein at, for example, pH 4.6, 20 degrees Celsius. You can get casein as a caseinate, and you can get whey proteins as soluble. You can get heat and isoelectric coagulation, where both of them coagulate, that's caseins and whey proteins. Or you can use thermal coagulation, where you then get, again, whey proteins and caseins as a mixture. Now, the target in industry, and possibly also in academia, is to ask ourselves a question: which of these proteins in this mixture might be better in terms of functional properties, and even in terms of bioactive properties? And if there is a protein that is not, can we get rid of it or reduce it so that we enhance the purity of the protein that we want? So the question that we get here is, we see whey proteins come in, and there are different purities, and whey proteins, especially made of alpha-lactalbumin and beta-lactoglobulin. The question is, what happens if we want either of them in its pure form? And there are methods which are available. But before we get to those methods, let's get a brief summary of what these two proteins, for example, which constitute whey proteins, what they are. And what we get here, you can put your finger on only two things, apart from all these stories. One, the two proteins are closely related in terms of their molecular weights. 14,000 Daltons for alpha and 18,000 Daltons for beta-lactoglobulin. The second thing is that their PIs are very close to each other, between pH 4.2 to 5.5. That means these two methods, the PI and the molecular weight is a very poor criteria for separating these two. So one of the things that is now being done is to exploit, for example, the instability of either of these proteins, either in the presence or absence of calcium or using heat. And we see an example here where whey protein beta-lactoglobulin is purified because of its sensitivity or insensitivity to, for example, TCA. By using TCA, somebody called Cheison in 1997, for example, was able to separate whey protein beta-lactoglobulin, which is soluble in TCA, from the other whey proteins. And that was done on the right side of this figure by combining TCA precipitation with membrane fractionation. So whatever was a pellet here is what could not dissolve. And beta-lactoglobulin was successfully purified as the end product. However, they could only obtain about 60% of beta-lactoglobulin, and the purity was relatively high, 95%. On the left side is work which was done here in Germany. These people used about 3% TCA, precipitated the other proteins, got beta-lactoglobulin, and then they introduced pepsin. Pepsin digests alpha-lactalbumin, but beta-lactoglobulin is resistant. It's not digested by pepsin. So what they did here on the left, you can see they added pepsin. That means to remove the extra alpha-lactalbumin. And they were able to, in combination with membrane technology, also get about 68% beta-lactoglobulin with a higher purity only by 3% of about 98. This is using TCA to get rid of the other proteins, purify beta-lactoglobulin. Let's go to the second method which is available, that is use of ammonium sulfate. This is a classical method. All proteins mostly are insoluble in 40% ammonium sulfate. When they used sulfate precipitation, Lozano and group recently, about 4 years ago, were able to adjust the pH, manipulate with ammonium sulfate, precipitate what was not required, that is the proteins that are not including beta-lactoglobulin, redissolve the protein, adjust the pH again, use ammonium sulfate, and that eventually led to obtaining beta-lactoglobulin, very low amounts of beta-lactoglobulin. From about 3.5 liters, they could get 2.09 grams. What we see here, the yield was very low, 14% beta-lactoglobulin. The purity was very good, 95. So these are methods which exploit salt sensitivity of the other proteins and the ability of globulins to dissolve in salt. That's the story of how beta-lactoglobulin is, for example, manufactured. From the presentation by Jose Toro this morning, lecture number 5, I think slide number 2, you saw how heat and addition of calcium and citric acid was manipulated, for example, to precipitate out alpha-lactalbumin and separate it from beta-lactoglobulin. So that is another method which is well established now, but I will not go into that. Let's look at how alpha-lactalbumin is, for example, fractionated, which goes back to that lecture. Because of the sensitivity of calcium chelation, if calcium is removed from alpha-lactalbumin, it becomes selectively precipitated. And somebody has used this method, quite a number of them, including our group also. Professor Kulotsik's chair has done a lot of work in this way of precipitating alpha-lactalbumin by manipulating the calcium using citric acid, heat combination, and membrane technology. And what we can get here is a product which is either about 85%, 86% alpha-lactalbumin recovery, purity of not so high, 75%, and you can also get a mixture of alpha-lactalbumin and beta-lactoglobulin. What we see from this method is that the method can be used to obtain both alpha and beta-lactoglobulin proteins. Okay? The second thing is, it is a process that depends on certain variables. How is the pH, for example, going to affect the process? The concentration of the protein also in the solution. Lactic acid or lactose, for example, temperature, the holding time during heating, and such other variables. And one of the things that we see here is that this process is prone to denaturation of the proteins to one degree or the other. There is a variation of the degree of denaturation of the protein. So that means we do not get a protein that is completely undenatured. And the other thing is there are definitely questions of yields. So this is with alpha-lactalbumin. You can get both by manipulating the salt sensitivity and precipitating out alpha, combining it with membrane technology and temperature, for example. Let me just summarize it for you in this table, which I don't need to go through because I think you already can access it. People have used TCA solubilization at low pH, and I give you here on the right side the references that you can refer to. What we see here is there is a combination of salt sensitivity, there is a combination of use of pepsin, for example, to digest alpha-lactalbumin. There is a combination of ammonium sulfate. There is also a different, a combination of different types of chromatographies, ranging from gel and anion exchange to affinity chromatography. And also some people have used expanded bed adsorption with hydrophobic interaction chromatography, which we will mention slightly later. Now, what do we see with these methods, in summary? That you can get from milk, you can start with milk, you can separate the caseins and the wheys, and then you can work with the whey proteins using different pathways that are summarized here to get alpha-lactalbumin, for example. Some of them give you both alpha and beta. Other methods only give you alpha-lactalbumin, while beta-lactoglobulin is not necessarily the target. Included here, and we will note this on the right side, is whey proteins digested using trypsin by Conrad and Kleinschmidt, and they could obtain alpha-lactalbumin. This is a method which is going to be developed further. What do we see with these methods that have been mentioned? Remember, there are salts which are used. There is denaturation of one protein or destabilization of the other. The protein yields are variable. We could see from literature, for example, that the yields ranged from 11 to 80%. We could see from the literature that there was protein denaturation of different amounts of denaturation reported or degrees. The conditions are very harsh. Some of them involve use of salts, and those salts are not very good for consumers. Nobody wants to eat a product which has a lot of salt, however healthy it is. We want a bioactive product, not a bioactive poison. And the proteins that are obtained are also of variable purities, from 60 to 98%. That's what we saw. And industrialization of methods like chromatography, however beautiful they could be, is really a challenge. So the question here, and this is what my group keeps asking, is there a remedy? And can enzymes provide a solution? And yes, enzymes work under mild pH and temperature. You don't need any additives for enzymes to work. There is low salt content at the end of it. You have minimum side reactions which are associated with high and harsh temperature, and you have total mass conservation. Nothing is lost. The protein you don't want is converted to peptides, total nitrogen conservation. And finally, enzymes are generally regarded as safe. That means they don't harm you. At least if you use the industrial enzymes, then it becomes a different problem. But if you use the food-grade enzymes, then they are safe. Now let's go to a brief background. You have whey proteins, which is a mixture of, for example, beta-lactoglobulin on the left, what you see here. On the left is beta-lactoglobulin. We simply highlighted for you the red, which indicates areas that we expect trypsin to cut, because of what we know theoretically trypsin to do, to cut between lysine and any amino acid and arginine and any amino acid. That's what is highlighted red. On the right side is alpha-lactalbumin. Also highlighted are the areas that we expect trypsin to digest. But we put our finger on two things. On the left side, beta-lactoglobulin is resistant to pepsin. And alpha-lactalbumin on the right side is resistant to trypsin. This is what we get from the literature. Now, these two proteins come mixed. We mix them with an enzyme in a reactor like this. What do we expect? Let me take you briefly to our hypothesis. Our hypothesis says if we have these proteins, which are there in whey proteins, for example, alpha-lactalbumin here, which we got from an RPH-PLC chromatogram, and beta-lactoglobulin exists as two genetic variants, A and B, which only differ by 2 amino acids, but sometimes that difference has a high impact on things like heat stability and susceptibility to even an enzyme like trypsin. So Conrad and Kleinschmidt, the paper that we saw before, have worked before at pH 7.7, 42 degrees Celsius. What they did was they took defatted whey, ultrafiltrated it, added trypsin and digested the protein to a DH of 9 to 10%, and then they did diet filtration and obtained beta-lactoglobulin, alpha-lactalbumin through spray drying. What they got here, if you can see on the right side, on the left side, yes, was that at the beginning of digestion to the end, there was literally less change in the amount of alpha-lactalbumin that was present in the reaction mixture. Whereas beta-lactoglobulin reduced to 0 in more than close to about 200 or 180 minutes. That's about 3 hours. So beta-lactoglobulin was being digested, alpha-lactalbumin was resistant, the amount of peptides was increasing. And we also see here that serum albumin was relatively untouched. But they could only recover 15% of alpha-lactalbumin, even though the purity was more than 90%, which is very good. So what do we know about beta-lactoglobulin from theory? That if you adjust the pH from pH 2 to around pH 9, beta-lactoglobulin changes its molecular conformation from a monomer through a region in the pH range of 2 to 3.7, where it exists as a dimer, that means two molecules together, and in the region of between 3.7 to 5.1, that's the PI, where especially beta-lactoglobulin genetic variant A exists as octamers, 8 monomers together. And then in the physiological region of milk, it exists as a dimer. And there is a critical red line here, which we underline. By pH 7.5, if you raise the pH above that, the dimer, which exists in milk, breaks to monomers. The protein separates into single molecules. And if you exceed pH 9.7, the protein is irreversibly denatured. If you put your finger on two things from this figure, number one, the pH 7.5 above that, the protein changes from dimer to monomer. And if you look at the arrow again on this side, it says above 40 degrees Celsius, the protein changes from a dimer to a monomer. We hypothesize that the hydrolysis environment is a major driving force. How? It impacts on the protein structure and its conformation. It also impacts, therefore, on how the protein interacts with the enzyme. For example, if we introduced an enzyme to this protein at these different pH values, the way the enzyme will attack beta-lactoglobulin at its octameric form of pH where they exist as 8 units, will be different from when it is a single molecule. Let's have an example. Start on the right side, top right. This is when we digested whey proteins using trypsin at the optimum conditions for trypsin, pH 7.8, 37 degrees Celsius. What do we see? Two things. Number one, alpha-lactalbumin remained a little unchanged, if you could allow that. It was digested, yes, up to around 44%. Beta-lactoglobulin A and B was depleted by around 40 minutes. At 37 degrees Celsius, 7.8 pH, alpha-lactalbumin could not be completely digested by trypsin. Beta-lactoglobulin A and B was completely digested. Let's move to the top left. When we adjust the pH to 7.8, but we do not touch, we don't touch the pH, but we change the temperature to 25, sink the temperature down. Alpha-lactalbumin remained relatively higher. Beta-lactoglobulin here is digested in a genetic variant dependent manner. If you see the one on the left means it's digested faster. Beta-lactoglobulin A was digested faster than beta-lactoglobulin B. So what do we see from this figure? Changing the temperature down, we could conserve about 70% of alpha-lactalbumin by removing beta-lactoglobulin. If we double the temperature from 25 degrees Celsius to 50, bottom left, we see that in 5 minutes, all the proteins were digested, except 9% of alpha-lactalbumin. In 10 minutes, there was no more of alpha-lactalbumin and no more of beta-lactoglobulin. So you can put your finger on one thing. If you sink the temperature from the optimum to 25, you increase the resistance of alpha-lactalbumin, you increase the selectivity of the enzyme. If you raise the temperature to 50, all proteins are digested by the enzyme similarly. The selectivity, the resistance of the proteins is lost. So this is how it looks like when we do HPLC. Alpha-lactalbumin, if you remember where it was, we could not get any more beta-lactoglobulin. However, we generate peptides which move to the left of the chromatogram from alpha-lactalbumin. That seems to be suggesting that the peptides from beta-lactoglobulin are less hydrophobic than beta-lactoglobulin itself. Let's summarize this. What you see here is a combination of temperature, pH from 7 to 7.7, which is a reference for us. And you could put your finger on saying pH 7 was very poor because all the proteins could still be detected. Alpha-lactalbumin at 25, the dark red one is 30, blue is 37, green, if green is visible, is 50 degrees. The best condition here was here, pH 8.5, where we could get rid of beta-lactoglobulin A and B at all temperatures. Whether at 25 degrees, 30, 37, and 50, beta-lactoglobulin was all gone, but only alpha could be detected at pH 8.5. If we worked at a higher condition, we are losing alpha-lactalbumin. Look at the green here. The green here is less than here, and it's less than there. So increasing the temperature and the pH led to more depletion or removal of alpha-lactalbumin. And if we used a lower temperature, for example, of 25 degrees Celsius and a higher pH, we could conserve close to about 68% of the original alpha-lactalbumin. And all beta-lactoglobulin was gone. So selectivity is improved here by, remember again, if you put your finger on that map, changing the temperature to above 7.5, you break the dimers to monomers, the enzyme can penetrate the protein more and digest it more, beta-lactoglobulin is gone, alpha-lactalbumin is resistant. Changing the temperature lower increases the selectivity, the enzyme can only digest beta-lactoglobulin and less of alpha. Changing the temperature higher, all the proteins are digested together. We did something which is rather new. We said, okay, trypsin was reported before. What we only did was to change the milieu. Say, okay, if we switch the conditions, does it affect anything? And what we know from theory is trypsin and chymotrypsin. These are two related enzymes. They share 40% of the amino acid sequence, including the triad. The catalytic pocket is conserved in both. Does chymotrypsin digest whey proteins the same way that trypsin does? This was the question. And what you see here is at 25 degrees, 37, and 50, work which was done by a colleague who came from Croatia. It showed that at 25 degrees Celsius, the proteins were, the selectivity of the enzyme was higher. It could digest beta-lactoglobulin, and alpha was resistant. And if you increased the temperature to 37, all the beta-lactoglobulins were gone. Some little alpha could remain. At 50 degrees, the enzyme was digesting everything. So yes, chymotrypsin is behaving like trypsin. As we increased the temperature, there was low protein recovery and low resistance. Alpha-lactalbumin was more resistant than beta-lactoglobulin, and chymotrypsin digested the proteins just like trypsin did. The optimum again was in the region that we found for trypsin, which is pH 8.5 and 25 degrees Celsius. The biggest problem usually is if you apply an enzyme, how do you stop the reaction? And there are 4 or so classical methods. Adjust the pH HCl, heat to denature the enzyme, use inhibitors. What you see here, BBI and EWI, are the Bowman-Beark inhibitor or the egg white inhibitor. When we did heating of a sample that was hydrolyzed at 25 degrees Celsius, look at what happened here. As the temperature rose from 25 to 80, the protein was being digested as the enzyme went through the optimum. It was better to use acid and the inhibitors than to use heating. That's the message you can get from here. So the question now is, how do we summarize this information? On the left, Kleinschmidt and his group did it at pH 7.7, 42 degrees Celsius. They could get alpha-lactalbumin through membrane filtration after hydrolysis with trypsin. What we did in our case was to say trypsin yes, but pH 7.7 no, temperature of 42 no. We sink the temperature to 25, raise the pH to 8.5. And we could see here 8.5, 25 degrees. We could use trypsin and chymotrypsin. With HPLC, we can confirm that it's only alpha-lactalbumin which is present in 2 hours. And at the moment, we are busy thinking about downstream processing. Why downstream processing? Because this is the biggest headache. We have succeeded. The red is showing you what we had at the beginning. We have beta-lactoglobulin, we have alpha-lactalbumin. After digestion, the blue, there is no more beta-lactoglobulin, but there is alpha-lactalbumin, there are peptides, there is enzyme, and there is inhibitor. How do we get rid of them? So that's why we are thinking about the downstream processing. And let me take you to the third example and final one, which is to say we know already that if you digest whey proteins using pepsin, pepsin will attack alpha-lactalbumin while beta-lactoglobulin is not digested. Does any other enzyme working under acid pH attack alpha-lactalbumin? We took a classical enzyme called acid protease A, which works at pH 2.0, and we mixed whey proteins with this enzyme, and this is what we saw. At the end of 120 minutes, we could see beta-lactoglobulin was almost intact. But alpha-lactalbumin was gone in about 45 minutes. So yes, acid protease A, like pepsin, digests alpha-lactalbumin, but beta-lactoglobulin is intact. And if you allow me, then I could show you that this is where you can get those results. They are published. Begin with whey proteins. If you go to the left, you use trypsin, you get that paper in International Dairy Journal. If you go to, in the middle, you use alpha-chymotrypsin. The paper is now online, Journal of Dairy Research. And on the right side, we have purification of beta-lactoglobulin. Fortunately, we still see beta-lactoglobulin A and B, but we don't see any more alpha-lactalbumin. That paper is now published in LWT. In summary, normally I speak too much, but today we are lucky. In summary, beta-lactoglobulin is digested by both trypsin and chymotrypsin if we only sink the temperature and raise the pH. The pH breaks the dimers to monomers. The temperature provides a stress on the enzyme. And when the enzyme is stressed, it digests beta-lactoglobulin. It doesn't digest alpha-lactalbumin. The conclusion from the acid protease A is that if we digest whey proteins in low pH, the enzyme attacks alpha-lactalbumin, whereas beta-lactoglobulin remains largely intact. You don't need to begin with whey proteins. If beta-lactoglobulin is an allergenic problem to your consumers in the dairy industry, You can simply take trypsin, pump into your milk, let it digest beta-lactoglobulin, you produce beta-lactoglobulin-free milk. Let me invite your questions. Thank you.